Hi, I'm Sebastian Springer and I'm a biochemist.
I'm Susanne Illenberger and I'm a biologist.
Together we teach biochemistry
and cell biology at Jakobs University Bremen in Germany.
In this course, we would like to share with you our fascination for one of the most beautiful molecules in nature, deoxyribonucleic acid, or DNA.
DNA is the keeper of genetic information in all living cells. But surprisingly, it is made up of only 4 building blocks. Now, the combination of these building blocks, one after the other, holds the genetic information. It defines individual identity, but also gives rise to the wonderful diversity of organisms in nature.
So we'll explain to you how this genetic information is maintained and how it is put to use in living cells. And together we will look at some classic experiments that have helped researchers to understand just how DNA is structured and how it works.
And with this knowledge, scientists have developed great techniques for the diagnosis and therapy of diseases, which often arise from alterations in the DNA molecule.
So our course is organized into 14 units of about 15 minutes each. At the end of each unit, there are going to be questions and assignments so you can test yourself. As we go along through the course, our assignments will become more and more complex. But don't worry, you'll be able to discuss them with other people in an online forum.
Now, of course, you'll ask yourself, what do I have to know to join in? You definitely do not have to be a trained biochemist or a biologist, but some high school knowledge about chemistry and biology will certainly help. To give you an idea, we will give you a short glimpse on what to expect.
So come join us on our journey from DNA structure to therapy. This here is a model of a DNA molecule, rotating so you can look at it from every direction. You can see it looks like a spiral staircase. The molecule has 2 backbones wound around each other. You can see them here on the outside. On the inside of the molecule, the horizontal steps of the staircase connect the 2 backbones with each other. Now let's assume we could unwind the DNA and spread it out on a sheet of paper. Then it looks like a ladder. The backbones or rails of the ladder are on the outside of the molecule. Now on the inside, one can see that each step is made of 2 parts, each one connected to 1 backbone that meet in the middle and fit together nicely. As you can see, red always pairs with green and orange always pairs with blue. These half steps are the 4 different building blocks of DNA that we mentioned before, and this is how they fit together due to their chemical structures that we will discuss in the course. This structure of DNA, with 2 backbones on the outside and building blocks pairing in the center, is just perfect for passing on the genetic information when a cell divides in 2. The DNA molecule first divides into 2 strands, as you can see here. Then each strand is completed to give the original molecule. Now we have 2 identical DNA molecules that can be distributed to the 2 daughter cells. Isn't that amazing?
Unfortunately, no system is perfect. Sometimes the sequence of DNA building blocks becomes altered. Such changes are called mutations, and they may lead to severe diseases just like cancer. Now, we'll look at some of these diseases and how scientists are working on therapies to cure them.
Curious? Then stay tuned.